Welcome everyone. In this chapter, electrophoresis and chromatography. We are going to study electrophoresis. We will learn electrophoresis and chromatography. The topic is electrophoresis principal force factor type application. Separation serum protein and agar gel chromatography classification pattern chromatography procedure. Other techniques observation TLC, gas liquid ion exchange, gel filtration, affinity, HPCl, LC. Electrophoresis introduction electrophoresis laboratory techniques used to separate charged filter protein and DNA based other movement in the electrical field. Electrical foundational technique like using the applied electric field, molecular move in different data based size, shape, and net charge. The principal electrophoresis charged molecule move in the electrical field. Partially charged particle move into a negative electrode, cathode, and anode. The speed movement depends on size, shape, and charge. Movement depends on charge in molecule, size of molecule, shape of molecule. Forces involved. The electrical forces main division force, vertical force. Factor affecting electric force, voltage applied, nature of buffer solution, size and particle, temperature, type of supporting medium. The type of electrophoresis, paper electrophoresis, gel electrophoresis, capillary electrophoresis. The gel electrophoresis acrosal and polyclimate. The application separation of serum protein, DNA fingerprinting, diagnosis of disease, study of the M-shape. The serum protein separation agar gel electrophoresis. The blood serum based on agar gel electric current applied, protein separated into the band, albumin, globulin, beta, alpha, gamma. Force involved in electrophoresis. The movement molecule depends on three mains. Electrical force division force versus charged particle through the medium. Further force of resistance opposite movement due to cell buffer. Gravitational force minor efforts usually negligible for lab condition. Factor affecting electrophoresis. The separation of molecule depends. Charge molecule more charge factor movement. Size of molecule smaller faster. Shape of molecule compact to shape of move faster. Sense of electrical field. Type of supported medium acrosal polyclinal gel. pH and ionic strength of buffer. Temperature high temperature may affect the result. Type of electrophoresis. Main type of include the acrosal electrophoresis used for DNA and RNA separation. Polyclinal gel electrophoresis PAGE used for protein. Serum protein electrophoresis separate blood protein. Capillary electrophoresis uses thin type of separation. Human electrophoresis compared antibody and electrophoresis. Acrosal electrophoresis used of DNA and RNA separation. The separated DNA or RNA molecule by size. How to work? Nucleic acid microtube through the porous acrosal matrix toward the positive electrode anode. Visual picture. The transparent horizontal gel matrix placed in the buffer tank. Under ultraviolet light, DNA or RNAs appear bright, glowing band, usually pink or blue, depending on the strain, such as Ethereum bromide. The smaller fragment travel further than the large one. The ultraviolet transmission lab documentation. This slide shows the power supply, the DNA fragment, the separation, DNA sample loading, Agrozole mixed the plastic trade. The supply the 150 voltage. The DNA sample load in the buffer. DNA third one DNA separation due 30 to 45 minutes. DNA sample marker the DNA slightly ultraviolet transfer documentation. The prepare gel acrosol powder mixed the electrophoresis buffer to the displayed concentration of heated in the microwave mild. The concentration of acrosol, the percentage acrosol depends on the size of fragment resolved. The acrosol refers the percentage acrosol volume buffer length acrosol gel normal in the range of 0.2 degree to 3 degree. Lower concentration acrosol faster DNA fragment migrate. The aim of separated DNA fragment, low concentration acrosol should be used in the separated small DNA fragment, high concentration of acrosol recombinant. Epistelim promote the added the gel final concentration 0.1 facilitated utilization of DNA after the electrophoresis. 60 degrees provide that calcium dry content the sample come allowed to the solid at the room temperature. After the gel solidated, comes the removal tank to reach the bottom of the well. The camper and cover the buffer. Pepper the sample well will the power relay the place upper the current applied. The observation bubble coming out of electrode. Positive electrode is usually the colored red with a negative range. The DNA migrated in the gel can suggest to visibly monitor migration of the tracking dye like promopenal blue and hexagonal light. The application electrophoresis. Electrophoresis field is used in biological and medicine. Separation DNA fragment genetic studies, protein analysis in the blood serum, diagnosis of disease, and DNA fingerprint in the foreign chain, genetic research and biotechnology, diagnosis diseases like liver disorder, kidney disease, and the infection. The polycarbonate PHE used in the protein. The PHE is electro technique that is used mainly for protein analysis, separation, protein nucleic fragment. Smaller than 100 pp and polypeptide chain depending on the electromagnetic. The biological biochemistry dentistry for the chemistry of biotechnology usually molecular biology. STS-PHGE of protein. There is a electro technique that separates polypeptide chain according to their molecular weight. Contains the sodium sulfate added to system order prevent denaturation condition. The polycarbonate gel condition sodium sulfate the sample with cancel the affection internal electrical charge sample protein. The separation becomes a large depending on the molecular weight of sample protein. STS-PHG and can be divided into the stocking and gel separating gel. The top of separating gel after the solidification and the gel pump comes inserted in the stocking the gel. The percentage shows according with size target protein in the sample. How does the electrophoresis pH work? The main principle polygel electrophoresis is separated molecule protein polypeptide nucleic acid passing through the process of polycarbonate using electrical current. The pH is additional ammonium persulfate. The reaction catalyzes tetramethylaminide from the net surface of analytic moving. Agreement includes the gel, smaller, the pore size, sense of smaller protein ability to pass through. The bicarbonate also impacted both sides, but often they kept constant. The speed which a small protein are able to move through the gel, improve their reduced solution, preventing the running into buffer repeatedly through current is applied. Application of acrosol electrophoresis DNA analysis used a separated fragment size, help in checking the result of DNA digestion, retrication, enzyme analysis, used a PCR protection verification. Another use of genetic fingerprint, helping the DNA profiling foreign change, used identified interval screening investigation and pattern testing. RNA analysis, used to study RNA integrity and size, helping the gene expression and studies. The molecular biology research used cloning experience of confirmed inserted gene, helping recombination DNA technology, medical diagnosis, the disorder of analysis DNA mutation, the identify infection like virus through the genetic material. Another quality check of biotechnology, check the purely size of DNA RNA sample before further the experiment sequence. Serum protein electrophoresis. Serum protein electrophoresis laboratory techniques used to separate proteins into the serum based on their movement to the electrical field. The sample explains the work. Blood sample placed in gel, electrical current applied, protein moved a different shape between size of charge, separate into the distinct bond. Main protein fragments show albumin, alpha 1 globulin, alpha 2 globulin, beta globulin, gamma protein, cloversamine. It is important help in the diagnosis disease, liver disorder, kidney disorder, infection, immune system problem like multiple myeloma. That's the slide show a normal protein electrophoresis, the albumin alpha 1, alpha 2, beta, gamma globulin the structure of it. Alpha antitrypsin HCl, DGP, alpha lipoprotein, hepato another one, beta 2, alpha 2 macroglobin, chloroblasmin, hepatoglobin. Second beta 2 gamma 2 hemoglobin, transferrin, beta lipoprotein. Application of serum protein electrophoresis. Serum electrophoresis is a laboratory technique used to separately study protein in the blood serum. It is mainly used in medicine diagnosis. The main application detection of multiple myeloma. The normal monoclonal protein M protein produces blood cancer. Diagnostic liver disease. Condition of hepatic change of protein level. Kidney disorder, urine related disease, nephronic syndrome. Inflammatory infection disease. The slower increase the decrease of protein fraction during the infection of inflammation. Immune system disorder, immunoglobulin disorder, autoimmune disorder. Nutritional status of assignment. Low protein level may indicate malnutrition and malabsorption. Capillary electrophoresis PC. The capillary electrophoresis laboratory technique is very charged molecule like protein, DNA, ion, molecule principle. The protocol move into electrical field and different protocol move to different speed up depending their charge, their size, the type of buffer solution. The capillary means a capillary very glass and plastic tube, almost like hair-like tube. The sample is placed inside the tube filled the buffer with the buffer. How separation happen? The small sample is injected capillary, electrical field applied through the tube of a different speed, separated into band the how to the first travel, detector record separation compound. The capillary involved reservoir electrode to buffer, EDF, detector to beta equilibrium, capillary outside the electrolyte buffer. The two very narrow residence. Voltage are applied to the safely. Through separation happen to the efficiently. DNA and RNA analysis, protein separation do a track testing, clinical documentation of uses. Immunoelectrophoresis laboratory technical component electrophoresis and immunological antigen reaction separators identify protein separate blood serum. Principal separating electrophoresis based on charge and size. Antibody added the gel, antigen protein antibody moved towards either the meet optimal proportional by the visibly precipitation line. Detection and identification serum. Disorder abnormal protein monoclonal immunoglobulin study in the antibiotic antigen reaction. Sample separated and used antibiotic identified specific protein by forming visible lines. A sample diagram shows a 2-mark examination question. Immunoelectrophoresis diagram such as gel tank and gel across classical power supply to disinfection gel neatly the introduction to vein blood well analyzed by blood sample. Immunoelectrophoresis diagram. It occurs electrophoresis to range level. The diagram is calculated the concentration unknown antigen. Produces immunoelectrophoresis. The gradual preparator slide the horizontal position. The application does carefully sample template 2.3 protein dilution solution 100 antigen solution plus 10 microneedle. The applied the cross the corresponding slide control the slide the sample slide. The electrophoresis camper the sample to cathode side the electrophoresis range for 20 means and 100 voltage. The electrophoresis computer corresponding under serum and the most chamber indicator 18 to 20 hours room temperature horizontal position. The horizontal position and try the blood slide. 10 minutes of trying the watching the repeater to skin. 70 degrees main do protein solution for about the main this coloring gel for 5 minutes this staining solution bath. Try the result the evaluation. Result of immunoelectrophoresis Professor precipitation present of antigen antibody interaction formation of precipitation suggests of no reaction. The identified pace of intensity, shape and position of the precipitation line. The application of immunoelectrophoresis identifies approximate quantity whether protein represented the serum. Protein identification immunological sequence of monoclonal and polyclonal gamma pathology. Abnormal proteins and myeloma proteins in the human serum. Protein mixture of contains a different antigen. Secreted protein separator begins observed hypogammaglobulinemia. Method of usually monitor antigen antibiotic. Single antibody mixture of antigen. Electrophoresis are older method of quality analysis. M protein serum medium. Diagnosis evaluation of the therapeutic response treatment disease state affects the immune system. Advantage immunoelectrophoresis. Electrophoresis powerful analytic technique, high resolution power of combination with immunodeficiency against anti-serum. Electrophoresis member of antigen identification in serum. Limitation of immunoelectrophoresis. Slower, less sensitivity of more difficult interpret and immunofluorescence electrophoresis. The M protein of mostly rapidly makes immunoglobulin present in the higher concentration may observe presence of small M protein. The food analysis limited the availability specific antibody.